Okay, good morning, everyone. Before we get started, a note about that anatomy lab extra credit stuff. The questions were based on a previous session, and I know they didn't talk about exactly the same thing this time. So anything that wasn't in the memorial service, that first question, I think it is, go ahead and skip that. Just write your reflections. Sorry about that. I guess I should have written the assignment after watching the memorial service. So that's a lesson for all of us there. Okay, but go ahead and submit those for extra credit points in anatomy lab. Okay. So today we're going to get started talking about muscles. So we're going to start talking about skeletal muscles. Last time when we spoke, we were talking about the somatic nervous system, which is how we're going to activate skeletal muscle. We talked about that neuromuscular junction. Those are going to be our nerves coming into skeletal muscle, creating basically action potentials in our muscles. So we are going to tap back onto that knowledge a little bit of the way through here, but first we're going to talk about the structure of a skeletal muscle. Okay. So when we look at a skeletal muscle, we're going to see it in layers. So you've all seen some skeletal muscle now in the anatomy lab. So we are zooming in on a muscle body. So the pink part is actually called the muscle belly when you're in the anatomy lab. And that's what we're looking at here. So it's going to be surrounded by some layers. So it's going to be surrounded by layers of connective tissue. So when you look at a muscle, there's going to be a thin translucent layer surrounding it called the epimysium. And that's this here. Actually, I'm going to quick pull this out and show you a picture of it in a real body. Let's see, can I get out of here? Epimysium. So when we're talking about that, that's like this stuff around the muscle, right? So that thin filmy stuff, that's the epimysium, okay? I like real pictures, okay? Then we're gonna have something called the perimysium that extends into the muscle body. So when we're looking at the muscle, you're gonna see these thin little fibers within it. Sometimes I'll tell you to like look at the fiber direction of something. What we're looking at is these little muscle fibers. Part of the reason we can see those is we have this perimysium invading the muscle, dividing it into little bundles. Okay, so these little bundles are called fascicles. So a muscle fascicle is like this group, right? We have these little fascicles in these bundles. Okay. So bundles of long, skinny sets of cells are the fascicles. There are hundreds to thousands of muscle fibers within a fascicle. So we may be getting quite small when we look at these fascicles. So we'd be starting to zoom in under a microscope to see those individual muscle fibers. Okay. So then when we're looking at an individual muscle fiber, we've pulled out just one of these, right, from a fascicle. So we're like zooming in on one of those. Okay, so when we look at the muscle fiber, we're again going to have endomysium, or sorry, we're again going to have another type of mysium. Now it's endomysium. Okay, so epi was surrounding around the outside. Peri means like next to, so it's extending between the fascicles. And then endo means inside. So it's inside the fascicle surrounding the muscle fiber. And then our individual cells are going to have a plasma membrane, has a special name when we are talking about a muscle fiber cell. So it's called the sarcolemma. That's our plasma membrane here for our muscle fiber. And a muscle fiber, which is basically a muscle cell, has a lot of nuclei. So we call it multinucleated. So we basically think of it as like a bunch of cells that kind of developed together, but like became one cell, right? So here we see those many nuclei in our muscle fiber. We also have a special name for the cytoplasm of our muscle fiber. So if we're talking about a muscle fiber, the cytoplasm is now called the sarcoplasm. So we're going to see this word sarco a bunch when we're thinking about these muscle fibers. So that should be a hint that you're thinking possibly about skeletal muscle when you see that root. So at the cellular level, we're again going to have more divisions and more strands inside our muscle fiber. So now we're zooming in inside the cell. So we have the outside, our sarcolemma, that's our membrane here, right? Inside, we are going to have a bunch of myofibrils. So those are strands within our muscle fiber, okay? So our myofibrils are going to have these filaments, right? So we're going to be thinking a lot about these filaments in a second. Surrounding these filaments and these myofibrils, we are going to have a specialized version kind of the endoplasmic reticulum called the sarcoplasmic reticulum. And this is going to play a different role in our muscle fibers than it does in like your normal average cell that you learn about in your first biology classes, because the sarcoplasmic reticulum is specifically going to help us activate and contract the muscle. And we'll talk about that process a little later on. Okay. So our sarcoplasmic reticulum is going to have swellings where there's a bunch of calcium. So we're going to think about a bunch of calcium hanging out in our sarcoplasmic reticulum. Okay. And later on, when we talk about how we contract a muscle fiber, we're going to come back to that and think about that calcium, because that's going to be important for triggering contraction of our muscles. There are also tubules associated with the sarcoplasmic reticulum. So that's this green stuff. These are our T tubules or transverse tubules, and they are attached to the plasma membrane, to the sarcolemma, as well as to these lateral sacs of the sarcoplasmic reticulum. So we will often refer to them together, the lateral sacs from the sarcoplasmic reticulum, and then also the tubule running within it from outside the sarcolemma into the cell and between these parts of the sarcoplasmic reticulum. We'll often refer to that as a triad. So triad because we have like 1, 2, 3 things as part of it. Okay. So the myofibrils. This is the actual sort of like contractile structure of our muscle fiber. So our myofibrils are going to be made up of 2 proteins, so 2 different types of proteins stuck together in strands. So we're going to have thick filaments, and our thick filaments are something called myosin. So our thick filament, myosin. You see our thick filament is this sort of orange line in the center. Then we have our thin filaments, and our thin filaments are a protein called actin. So myosin and actin, our thick and thin filaments, are going to be really important for muscle contraction. So these different types of filaments are going to slide past each other to make our muscle shorter, to contract this muscle, to allow you to move. So what we're going to zoom in on next is a structure called the sarcomere. So the sarcomere is going to be a part of the myofibril. Basically, what we're going to do is we're going to chop this myofibril up. And we're going to take just 1 unit. So we're going to be taking like these little chunks. These are our sarcomeres. So I'm chunking up this myofibril into individual sarcomeres. So that's what we're going to zoom in on next. So our sarcomere under a microscope would have a structure that looks like a bunch of bands, right? So we're going to see a bunch of lines of different amounts of grayness, essentially. So these are the various bands and zones that we would see under a slide if we're looking at a sarcomere. They correspond to where we have our thick and thin filaments and how much of those. So we're essentially looking at zones of kind of like overlap, right? So when we look at the A band, so the A band is this whole area from this thick gray part all the way over to the end of this thick gray part. We have this dark band, and this is corresponding to everywhere that we have our thick filament. So our thick filament here is now this part. So again, it's this orangey part. So this line in the center of the sarcomere. going horizontally across, that's our thick filament. So anywhere we have that thick filament, that is going to be within our A band. Okay. Then we have our H zone, which is located within that A band. So here we have the H zone. Okay. So the H zone still has thick filaments. Okay. But this is going to be an area of the sarcomere where we have no overlap between the thick and the thin filaments. So actually, when we're talking about overlap, let me, on this zoomed in sarcomere, show you those thick and thin filaments again. Okay. So what we're looking at for our sarcomere, we're going from this zigzaggy line. This is actually called our Z line, or zigzag, right? So this Z line is basically the start of our thin filaments. So our thin filaments form this Z line, this zigzag kind of sewn together looking piece, okay? And so the other end of the sarcomere is going to be the next zigzaggy line over here. Okay. So these are made of thin filaments. Okay. Highlighting that. So that's the sarcomere from the zigzag line to this other zigzag line made of thin filaments. And anywhere we see blue on this image is also thin filaments. So we can see that there are projections towards the center of the sarcomere, right? So we have this projection. I'll draw an arrow on it to kind of highlight, right? So this projection, all of those are still thin filament, this blue stuff pointing in. Okay. So those are the thin filaments. And then our thick filaments are these things that look like wheat kind of inside. Right. Nope. Okay. So this stuff that we have in the center with these little heads or feet coming off of it, those like little wheat kernels, this is our thick filament. Okay. So when we're talking about overlap, what I mean is that we have regions where you have some thin filament and some thick filament, right? So like this would be a region of overlap, but this would be a region where we have only thick filament, right? And this would actually be a region where we have only thin filament, right? We don't see any of those little strands of wheat extending into this region. Okay. So when we're talking about overlap, that's what we're talking about overlapping. Okay. Sorry, so back to these things in order. Actually, let's do it from bottom up. That's easier. Okay. So those zigzaggy lines, we're gonna mess with the order. So those zigzaggy lines, our Z lines, are dark here. Okay. So then let's go in next. So if we expand, we're going to look for somewhere where we have no overlap with the thick filaments. So we're looking for where we have just the thin filament. Okay. And that forms our I band, where we have this pale area. And the reason it's called an I band is if you take this region on one sarcomere where there's no overlap and go all the way into the next one to its region where there's no overlap, you'll have that Z line in between, right? And so you can think of this as like looking like an I, right? So that's why this is our I band, okay? So then we have the A band that we were talking about. So the A band is anywhere we have thick filament, whether we have overlap or not. Okay, that's the A band. Then the H zone in here is where we have only our thick filament. All right, so only thick filament, no thin filament. Okay, and then the M line is just the center line. And the way I would remember M line, and actually this can help you remember which is thick and which is thin, M line for myosin. So myosin is forming our thick filament. This is the M line made up of myosin. So when we look at the stuff in cross section, we would have these things kind of layered on each other. We're going to be looking at it flat over and over again throughout our understanding of like the muscle physiology, but this is a 3D structure here. So we can see that if we cut it across, we have more thick filaments and more thin filaments than we just see on this kind of planar view. Okay. So this is what it would look like under a slide. So now we're going to talk about those strands and what they're made of. So we have our two proteins that we're interested in. The actin is the thin filament. The myosin is the actin is the thin filament. The myosin is the thick filament, which is why that line in the center is the M line connecting the myosin together. So right now we're talking about that blue stuff forming that zigzaggy line and the projections into our sarcomere. So this is actin, and actin contracts, or actin is one of our contractile proteins, okay? So actin is made up of a bunch of little subunits, our G-actin, right? So we have G-actin binds together into a strand, creating F-actin. Once we have all these little G for, I'm making this up, but G for globules, right? We have these globules of actin, creating this fiber of actin. So F is the fiber of actin, okay? And then we have two of those F-actins, so those fiber kind of things wrapping together to form a double helix. So our actin, when we're looking at our sarcomere, is going to be like two of these swirled around each other. So this is our actin strand, and we're going to have two other regulatory proteins that work with the actin to help us create muscle contractions. Okay. So on each of the little globules, on each of the G-actin, they look kind of like olives because they have this pit area, right? This like thing that looks like a hole. It's not a hole, it's a binding site for myosin. So we're going to have our other contractile protein. We're going to have our thick filaments. They have a spot where they can like grab on to the actin, and we will see in detail how that works. But this means that when we look at a strand of F-actin, right, we have a lot of myosin binding sites down it, right? So you could draw a line between these, okay? And when we are looking at our full complex, we are going to have something called tropomyosin that blocks all of these binding sites. So when you don't want to contract, you want to stop your different types of filaments from grabbing onto each other. So you need something to cover up these binding sites so they're blocked, so that you don't have myosin and actin sticking together. So that's what the tropomyosin does. So the tropomyosin is this like pink spaghetti covering over all those myosin binding sites. So that's the tropomyosin. But when you do contract, you want your actin and your myosin to work together. You do want to bind them together. You want to let them pull on each other. So you're going to need to uncover those binding sites. And that's the role of this little upside down snowman called troponin. Okay. So troponin is a complex of 3 proteins, and its role is going to be to push the tropomyosin aside. So to push that little pink strand to the side to uncover the myosin binding sites for when we contract. So the troponin complex has these 3 little pieces. It has an I part that attaches to the actin, has a T part that binds to the tropomyosin, and it has a C part that binds to calcium. Okay. So we can already kind of guess, right? We talked about the calcium in the sarcoplasmic reticulum. We know there's calcium there. We knew that in our neurons, the calcium kind of triggered neurotransmitter release, right? So this calcium binding in our muscles, we're eventually going to go through the process where this calcium allows us to create skeletal muscle contractions. Okay. So now let's look at the thick filament of the myosin. So this is our other sort of contractile protein. So we have these strands of myosin, these thick filaments, And what they actually are is like kind of like golf clubs sitting handle to handle, or at least that's what they look like to me. Okay. So their tails go towards the M line. So the M line would be here at the junction. So you can see this slight little gap here. I mean, it's not a gap because they're touching each other end to end, but they're not bound on here. Okay. So those tails towards the M line, towards the centerpiece, and then their heads, just like, you know, the other end of the golf club. I don't golf, sorry guys, is out towards the I band. So this is the end that would be out towards our actin, okay? So out towards the thin filament. So in the sarcomere, right, this is the part that's over towards this stuff, right? And we'll draw this on later, right? This will be in a future image, but this is where our heads are, essentially. Okay. On the myosin head, so this stuff on the head, we are going to have binding sites, and we're going to bind two different things. We are going to bind actin, right? So the reason we had a myosin binding site on actin is that we have an actin binding site on the myosin, and they kind of like magnet together, right? So just be careful when you're looking at these terms, right? The actin binding site is on myosin, right? So it's named for what it's going to attach to, not for the structure that it's on. And vice versa for our myosin binding site, right? The myosin binding site was on our actin because it's naming that we're going to connect to myosin there. We're going to bind to myosin. So that's one part on our myosin head. The other site that's important on our myosin head is that we have somewhere where we bind ATP. So we have our ATPase site. All right, so here's our ATPase site. And this is where we're going to be using energy to create skeletal muscle contraction. So this is actually why you need ATP in order to contract your muscle, in order to move stuff, is because we're going to bind our ATP onto this ATPase site on the myosin. We're going to break it up, get the energy, and use that to create skeletal muscle contractions. We will sometimes refer to our heads as cross bridges. Okay, so we could refer to these as cross bridges. The reason for that is, right, they're creating a bridge across to the actin, right? So they're going to bridge us onto the actin when we have the actin binding site bind to the myosin binding site. If we're looking at our larger sarcomere, right, we don't have just one thick filament. We don't just have one myosin strand. We're going to have them all arranged like this so that we have our cross bridges kind of at different levels, right? So we have cross bridges here, 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 and so on. There's going to be a central area called the bare zone where we have only tails, but that's relatively small. We have a lot of areas where we have lots and lots and lots of cross bridges, not just one. Finally, we do have one protein that kind of holds together the shape of the sarcomere. So this protein is not going to be involved in the contraction itself. It's not going to be shortening our skeletal muscle. It's just going to make sure that these fibers kind of stay where they're supposed to be. Okay. So this is the titin. Okay. And we didn't see that in our microscope slide. We didn't see that named in our bands, right? Because we don't focus on it, because it's not part of the contraction. It's just an anchor. And you can see that its structure is kind of springy, right? So this is an elastic protein, because what we're going to see when we do skeletal muscle contraction is we're going to see that our actin fibers are going to be moving in towards the center, right, relative to the myosin. So the myosin is kind of staying in place, but we can also think of it as kind of moving closer to the Z lines, right? So this titin is going to have to get shorter when our sarcomere is short. It gets longer when our sarcomere is long. So that's why it has that kind of springy looking structure. So we would say it has this elastic property so that it can kind of change its length just in response to the movement of the actin and the myosin. So it's just anchoring those thick filaments, which were connected at the M line, to the Z line, which is where we have all our actin connecting to itself. So now we're going to think about the actual force generation of the muscle. So we're thinking about how do we actually create that contraction, right? Like what is actually creating these things, this shortening, right? So the way we're going to do this is through something called the sliding filament model. So none of these proteins, other than the titin that's just kind of in that spring-like structure, none of our contractile proteins are actually changing their length. What's happening? I'm going to do this. Okay. So what's happening? This marker is my thick filament. My fingers are the thin filaments. What's happening is this. That's it. We're just having these things slide past each other, right? So our thin filaments are going to slide towards the center. So overall, this makes the sarcomere shorter. And that's it. Okay. This is called our sliding filament model. The sliding filament model, how the muscles move or contract. And it's the thick filaments and the thin filaments sliding past each other. The thin filaments sliding in because those thick filaments are grabbing on them and pulling them. Okay. So the thick filaments, that myosin, those cross bridges, those heads, are grabbing onto the actin at the myosin binding site. And they're going to snap their heads in and drag the actin, drag the thin filaments in towards the central line of the sarcomere. So force them to slide in, slide past them. And now our sarcomere becomes shorter. And as every sarcomere in that muscle fiber becomes shorter, the muscle as a whole becomes shorter. So that's what's happening with the contraction. Okay. We're going to go through the details of that process. So that's going to be something called the cross-bridge cycle. We're going to have a series of steps, kind of like we had a series of steps in metabolism, right, that allow us to take those proteins and drag those thin filaments in towards the center line. Okay. So that's our cross-bridge cycle. And eventually we will also talk about how we start that cross-bridge cycle, which is where we come back to that neuromuscular junction, right? Think about the nerves coming to the skeletal muscle and how those action potentials coming down are somatic nerves coming into the skeletal muscle at that neuromuscular junction, how they're going to turn on the cross-bridge cycle and create this contraction. So that's what the excitation contraction coupling is going to be. Excitation, referring to that depolarization that we have coming running down our somatic neurons. Right? Excitation, we have that charge basically, so that's the nervous impulse. And then we have our contraction part, which is the physical contracting of the muscle. And these two things are paired together. Finally, we'll eventually talk about muscle cell metabolism. So we'll talk about how the muscle cells are providing the ATP that we're going to see is needed by the cross-bridge cycle during contraction. So this is kind of a list of the topics that we're going to be looking at as we think about force generation. So we're going to start with the sliding filament model. So our muscle is contracting. What we mean when we say a muscle contracts is we mean it gets shorter, right? So we know that when it gets shorter, right, it's pulling its two endpoints closer together, pulling those origins and insertions closer together, creating movement. The reason it can shorten and lengthen is because of those regions of overlap. So I'm going to draw a sarcomere. I'm going to draw two sarcomeres. Okay. So I'm drawing on my Z line. I'm going to draw on those actin strands. Okay. So we are going to have a longer version here. Okay. Got my little wheat. Okay. Draw on here. Okay, so when we contract, we're sliding past. So that myosin is staying the same length. So I'll draw those again. Something the same length, trying. Okay. I should just use the line tool. Okay. So the myosin stays the same length. And what happens is the actin pulls towards the center. Okay. So now our Z lines, instead of being out here, they're going to be more like here. All right. All right. So we're pulling them in. But the overall length of this part doesn't change either. There's just more overlap now. So this is how we're creating a shorter sarcomere, right? We slide these past. So we only have a little bit of overlap in our longer sarcomere, but then we have a lot more overlap in our shorter sarcomere. So that's what I mean by we're sliding past, right? We're sliding those Z lines in towards the center. Okay. So if we were to look under a microscope slide, we could describe this as changes in the size of our bands, right? So the A band was anywhere we saw thick filament. The A band is anywhere we have myosin. The myosin did not change its overall length, right? It didn't like the protein didn't get shorter. It slid another protein past it. So the A band is going to stay the same length because we still have the same amount of myosin. The I band was referring to the Z line and then this area of no overlap on either side, right? This was our I band, okay? And because the sliding filament changes how much these proteins are overlapping, right? We're sliding the Z lines in, creating more overlap between the thick and the thin filaments. This means that that I band shortens as we have more areas of overlap. The H zone was the other area where we have no overlap, except the H zone was no overlap, and we have only myosin, right? So the H zone here was just thick filament, just myosin. Because we are sliding everything in closer together, that's also shortening as the sarcomere shortens. Okay. So this is kind of how they figured out what was going on in muscle contractions without looking at these individual proteins. Right? They didn't physically see these back in the day when they figured out how muscle contraction works. They looked at the changes in these sizes of these bands under a slide, and we're able to sort of work backwards to figure out how the physiology of that muscle is working. So next, we're going to be thinking about why we're sliding. So this is going to be about these cross bridges. Right? Our cross bridges were these heads on the myosin. So they are going to be forming bonds to the actin, essentially. Right? So they're going to be grabbing on, right? The thick filament to the thin filament, the myosin to the actin. They're going to be pulling it towards the center, and then they'll let go. Okay. So we're going to have a formation and breaking of cross bridges called the cross bridge cycle that allows us to do this pulling action. And here's just pictures of those bands. Okay. So when we have the cross bridge cycle, we're pulling our thin filaments in towards the center. So we're pulling actin in towards the center of our sarcomere. The way we're doing that is our heads on the myosin are grabbing onto it and dragging it, sliding those thin filaments past. Okay. And we sometimes think about this as like a rowboat, right? We think about these heads as oars paddling towards the center. So in that analogy, we're going to think about the cross bridge, so that head of the myosin as the paddle of the oar. Okay. Yeah. So the paddle of the oar, right? We're grabbing the water. Okay. The linkage to the thin filament, so the linkage between the actin and the myosin is the moment our oar hits the water. Then we're going to have something called the power stroke, as we pull our oar behind us, right? So moving our little boat forward. So we're moving the thin filament towards the center of the muscle. And then we pull our oar out of the water. This is the moment when the thick and the thin filaments detach. And then we reach back forward again to make another stroke with our oar. And so our myosin head is returning to the initial position. So this is the cycle as a whole, which is why we use that analogy, right? Because sometimes it can look a little overwhelming. So it's going on here. So we're starting, number one, down here. Okay. So we're starting here. Okay. So in the cross-bridge cycle, we start with the moment that the myosin and the actin bind together. Okay, so that's the moment our oar is hitting the water. We're binding together myosin and actin. All right, so we can physically see that binding. Here we see that this head, this oar, has popped out, and the actin binding site would have been here. Here's our myosin binding site, that dark spot. They're binding to each other. Okay, so that is the first step of our cross-bridge cycle. Right, our myosin grabs onto the actin. Okay. When this happens, we're going to have a shape change as this little phosphate ion gets released. Okay, we have a shape change here. And now, as we had that shape change, proteins change shape. So we now have our head flicking in, right? So it's gone from being out straight like this, and we just flipped it, right? So we flipped it like that from step one to step two. So that is our oar paddling through, right? So our paddle is straight, and now we just push. Okay, so that push is called the power stroke. So that's step two here. So step two is where the myosin head is bending in. It's bound to the actin, so that pulls the actin towards the center of the sarcomere. That's what this arrow is about, right? So we're pulling actin as we change the shape of this head here. Okay. Now we're going to be stuck for a little. So to do this, we release this phosphate ion. We're now in a low energy form as we also release our adenosine diphosphate, ADP. So we're going to have a step called rigor. Okay. So rigor as we release this ADP. So we don't have any energy. We can't do this again until we get more energy. So that's rigor. We already used up all our energy doing this power stroke. So after rigor, our myosin was in the low energy form. We bind on ATP. And we're going to return to the beginning, right? This is what this whole next half of this cycle is happening. So we have our low energy form. New ATP is binding to the head, right? So the ATP is going to bind on here. Whenever we have binding onto a protein, we're likely to have a shape change, and we do have a shape change here. The shape change is first the unbinding of the myosin and the actin. So when ATP binds into the ATP site, the ATPase site, unsticks these two proteins from each other. So now we have a gap. Okay. And then we are not yet back to the beginning. Now we take that ATP and we break it. So we break it, ATP gets hydrolyzed. So ATP gets broken into ADP plus a phosphate ion. Okay. Right, because ATP was adenosine triphosphate, ADP is adenosine diphosphate. So the T was for 3, the D is for 2. Okay, so we're breaking that ATP into 2 parts. Okay, and as we do that, we tuck this head back in, we tuck the cross bridge back in. Okay, so now we have what we call the cocking of the myosin head. So we're going into the high energy form. Okay, and that's readying us to go back to the beginning to bind myosin to the actin again. Okay. So we started with myosin in a high energy form where we have broken an ATP into the adenosine diphosphate and the phosphate ion, but we haven't used that energy yet. We then use that energy to propel the actin forward. Okay, so this is where we use the energy, basically, when we release this phosphate ion, changing the shape of the myosin molecule, pulling the actin towards the center. That put us into a low energy form where we can't do anything until we bind another ATP molecule, break it, and put us into our high energy form. All right. Take a second, explain this cycle to yourself in your head. I want you to pause, and I'm going to take a minute for you to think, and now is a good time for questions. Because I know this is a kind of complex process. As we're going through and as you're reviewing this, I'm going to just write down some questions that I think you should be able to answer as you understand the process. We'll go through questions like later, like practice questions, but you might be answering questions like this, right? Okay, so I'm just off my head looking at this picture and thinking about some things that this picture tells us. So as you are going through, what I want you to review in your minds and preferably on paper over the weekend is take a good hard look at that cross bridge cycle. Try explaining it to some friends. I don't know, explain it to your mom over the phone, something, and that's going to clarify for you which parts of this are extra tricky, which parts of this you don't understand. So on Monday, You'll have good questions for me, so I can help clarify.